Thanks, Elaine. Good morning, everybody. My name is Ross Macdonald. I'm the CEO of Cynata Therapeutics. Let me begin by thanking the ARM and the Sanford Institute for the opportunity to present to you today. It is a great pleasure to be here, my third year in a row. So let me tell you a little bit about Cynata Therapeutics. As Elaine indicated, we're an Australian mesenchymal stem cell therapeutic stem cell company developing a unique manufacturing technology based around induced pluripotent stem cells. I draw your attention to our safe harbour statement and remind you to check our website and the Australian Stock Exchange platform for announcements and other information about the company. A few key facts about Cynata. We believe our MSC manufacturing platform, which we call Cymerus, is a disruptive new technology, new generation approach to manufacture of MSCs. It's strongly protected by patents. It allows the economic and robust production of a clinical grade product. We have a number of strategic collaborations and commercial arrangements that I'll talk more about during our presentation. An experienced management team who's been there and done it before. Obviously, using iPSCs as a starting material, we don't have the ethical issues surrounding embryonic stem cell derivation. Low development risk, since we are essentially developing a manufacturing platform, and we're just about to commence a phase 1 clinical study. The company is listed on the Australian Stock Exchange. Market cap's about $34 million US, and we have around about 7 months cash remaining. Around about 1,900 shareholders across Australia and here in the US. The last year has been tremendously exciting for the company. We've attained a number of very important value accretive milestones, and I draw your attention to a couple of those in particular. The first is a recent announcement relating to a partnership we are developing with Fujifilm, one of the leaders, of course, in the regenerative medicine space globally. And on the bottom right-hand side of the screen, another very important announcement a couple of weeks ago was that the company had received clinical trial approval from the UK regulatory agency, the MHRA, to commence a clinical study in Graftversushost disease. Now, this is a genuine world first, a term that is often bandied around cheaply, but in our case, it is the first allogenic iPSC-derived product to enter the clinic. As many of you know, of course, Recon Institute started a clinical study a couple of years ago with iPSC-derived retinal cells, but that was autologous. And in this case, this is the first off-the-shelf product, in our case MSCs, but nonetheless the first for any iPSC-derived allogenic tissue. A few words about our partnership with Fujifilm, and that, of course, is a company that needs no introduction in this audience. We announced our relationship with them in the early last month, and that's a definitive agreement to enable Fujifilm to manufacture, market and sell our product CYP001 for graft-versus-host disease. The collaboration will involve the strategic acquisition of Cynata shares. They will become approximately a 10% shareholder in the company. There will be upfront milestone and royalty payments on product sales. And of course, Fuji has totally reinvented itself as originally a photographics product company, now to a major player in the regenerative medicine space. The company's, Cynata's therapeutic product pipeline, of course, is dominated at the moment by our phase 1 program that we hope to commence within the next couple of months in graft-versus-host disease. Low-hanging fruit for us commercially, but it's the final step to demonstrating the utility of our cells derived from iPSCs, as I explained before, in a clinical situation. We also have a number of other programs underway at the moment through academic collaborations, and they don't represent any surprises to those of you who understand the indications, the potential indications for MSCs. We also are working on an engineered MSC approach too, in a collaboration with Harvard MGH in glioblastoma, where our MSCs are modified as a potential cancer treatment, likewise with a German company called Apceth that I'll talk more about shortly. A few words about graft-versus-host disease. Obviously a fatal complication associated with bone marrow transplant. Not particularly commercially attractive. It's a relatively rare disease, thankfully, but nonetheless of great interest to those of us in the MSC space because, as many of you know, a product has been approved now in Japan for this indication, and product sales have commenced. The treatment costs are around about $100,000 to $170,000 per patient. So we've targeted that disease as our lead indication for our phase 1, and obviously successful results in that study will allow us to progress to much more commercially attractive but also more complicated clinical programs. So given the excitement that still is evident in relation to MSC-based therapeutics, why is it that we're not seeing much more progression of these agents in therapeutic uses? And that's because we believe there is a major roadblock to their industrialisation in the manufacture of these products. Those challenges are summarised in production scale-up, resulting in inconsistent product quality, reduced product efficacy, and of course the very important issues from a regulatory perspective of donor variability. The current manufacture of MSCs, as I'm sure all of you are aware, involves either a bone marrow extraction or isolating MSCs from other tissue sources such as adipose, placental tissue and so on. The yield is relatively low, and of course this requires massive expansion of those cells in vitro, which now the literature shows is a major issue because of senescence that occurs in as few as 13 population doublings. Now, 13 population doublings using 20,000 cells as your starting material from a bone marrow aspirate is only going to result in 1.6 doses. So right off the bat, we have a numbers issue here that results from the need to massively expand the finished product that either can only be solved through multiple donors, which has all the regulatory issues associated with it, or addressing in some way the clear issues of senescence and associated efficacy issues that arise from that massive expansion requirement. Of course, multiple donors lead to multiple problems, and any of you who have been at the regulatory presentations today and yesterday will understand the challenges that are associated with potency assays and comparability studies that are required in order to demonstrate that a change in donor is not going to result in a change in the key characteristics of the product and ultimately a different clinical outcome. And this is an issue which of course is convenient to us because we have a solution to it, but it is receiving considerable attention in the literature at the moment. And I draw your attention to a couple of commentators such as Katarina Leblanc from the Karolinska and Jacques Gallipeau, who have published extensively in this issue. So the challenge in summary is the requirement at the moment for either hundreds of donors per year, which is in our view not commercially viable, or massive expansion. And the alternative, the solution, we believe, lies in starting out with a completely different material, not relying on primary tissue sources as the starting material, but rather to rely on an alternative initial starting material, which in our case is induced pluripotent stem cells. And our technology, we believe, provides a very simple solution that is better, cheaper and faster in the manufacture of therapeutic mesenchymal stem cells. iPSCs, of course, have enormous clinical potential and we don't need to underscore that at this meeting, but our patented technology allows the expansion potential of iPSCs to be exploited in generating sufficient quantities of finished product, mesenchymal stem cells, for therapeutic use in widespread diseases such as cardiovascular disease, stroke, osteoarthritis, all of the diseases that are often cited as being the major economic challenges of our ageing populations. The iPSC expansion potential, of course, is well described in the literature out to 10 to the power of 72 cells, so an unlimited starting material is not a bold claim in our view. So in short, we believe we've taken what is essentially a cottage industry and commercialised it, much like the automobile industry was 100 years ago, where it was niche, a high level of human intervention, a very slow process, and resulted in inconsistent product quality, to the way the automobile in a modern factory is manufactured today, which is essentially robotised. Or, to use another analogy, 15 years ago we all carried a big fat Nokia product around in our pocket that made telephone calls. The next generation, of course, is the iPhone or the Samsung smartphone that makes a heck of a lot more than phone calls. So this is the evolution of technology that Cynata believes is going to be the future of MSC-based therapeutics. One of the important steps in the past few years for us has been to ensure that our product has the same characteristics of freshly isolated bone marrow MSCs, which in our view, and a view that's supported by many other commentators in the literature, represent the current gold standard. So we've undertaken analyses of cell surface markers, differentiation potential, immunopotency assays, genetic profiling, and ultimately conducted a number of in vivo animal model studies, including a study in critical limb ischemia, which is recently published, and a graft-versus-host disease model, which is just about to wrap up. The technology has come from the University of Wisconsin, so despite the fact we're an Australian Stock Exchange listed company, the origins of our IP are here in the US. In particular, Jamie Thompson and Igor Slukvin's lab, and of course Thompson has been often described as the father of stem cells, in particular, of course, with his appearance on the front cover of Time magazine a few years ago. So the technology has an outstanding pedigree and is well protected by patents from our licensor, the Wisconsin Alumni Research Foundation. Our business model, as I explained before, is to develop commercial partnerships. Cynata does not have a goal of becoming a fully integrated biotech company, but rather with an enabling technology, as we believe we have, our goal is to enable commercial partners to utilise that technology to develop MSC-based therapeutics for their particular target disease. For example, we have a relationship that is developing with Apceth, a German company that is looking at genetically engineered MSCs in cancer, one of the very, very exciting applications that have emerged for MSCs over the past few years. And of course, we have our growing collaboration with Fujifilm as well. This business model allows the company to derive early shots on goal, revenues in the near term by way of upfront and milestone payments, as well, of course, as royalty payments down the track. And ultimately, it looks to the potential for M&A, which, of course, in our space is always an active dialogue between small biotech companies and big pharma, with the acquisitions that have occurred in the past 12 to 18 months, notably CDI, the relationship that Apceth has with Helios, and of course the acquisition by Astellas of Ocata earlier this year. So the future for Cynata, we believe, is very, very bright. We expect to close the Fujifilm deal within the next couple of months. We will commence our phase 1 clinical trial later this year. The study centres are in the UK and Australia. We will complete the license agreement with Apceth to ensure that that company is enabled to develop our technology in their particular cancer indication. Obviously, we hope for continued success among the MSC companies, our brothers and sisters in the industry, because obviously any failure is going to result in a substantial re-rating of all MSC companies. So we look forward to those companies to continue to have success. And then finally, of course, the opportunities in engineered MSCs, in particular in cancer, is one of the key areas. So now we believe is the right time to invest in Cynata. The existing market issues associated with manufacturing, I've explained thoroughly, and of course the future potential for MSC-based medicines. It gets plenty of airplay. And our technology, we believe, overcomes the critical hurdle in that final step of industrialising this exciting therapeutic regime. So I'd like to thank you for your attention, and if we have a minute or two for questions, I'm happy to take those through the chair.
Can I ask one question?
Great. Thank you, everybody. Thank you.